When we talk about ourselves, right? First thing is first, let's first talk about uh how we observe ourselves. Okay. When we observe ourselves, so there are two types of structures that we can see within the cells. One type of structure is the structure that you can see using a simple microscope, right? That would include your nucleus, that would include your your cytoplasm, your cell membrane. All of these are structures that you can visualize, that you can see directly with a simple light microscope. Now, the way a light microscope work is that when we talk about light, so light has a certain wavelength. Those objects that are smaller than the wavelength of light, because we will study this when we do quantum physics in your A levels. When you do it, you'll study that light travels both as a particle and as a wave. So what happens is that the light has a wavelength. So when you talk about light, so those particles or those structures inside a cell that are smaller than the wavelength of light, you and I cannot see them using a light microscope, right? Those structures that cannot be seen using a light microscope, we call them ultrastructures. Now, before we talk about what these ultrastructures are, what are these structures that you need to know about in detail, let's first talk about how we can observe cells, right? One of the clear and easy way to observe cells is, of course, you can use a light microscope. It's you guys are doing your AS biology. You guys will be giving your paper 3 exams. You guys need to know how to use a light microscope. You guys are going to be in your paper 3, in your practical exam. They are going to give you slides. You are going to have to focus them. You're going to have to see them, and you're going to have to answer questions on them. So the way a light microscope works is like this. Okay. So we have at the very top over here, we have the lens, the eyepiece. We call it eyepiece. Eyepiece is where you're going to place your eye to actually look at your slide, whatever slide is given to the exam. To visualize it, you would put it in the eyepiece. Okay. Eyepiece has a magnification of times 10. That means whatever you are seeing, the eyepiece will magnify it by times 10. Now, later on, as you go down, you will have another lens. This lens is known as the objective lens. Okay. What does the objective lens do? So this objective lens, you can rotate it. You have got multiple options. Generally, we have, for example, 10 times, 40 times, 100 times. So you have got lenses with different magnification. Your laboratory in your school might have different magnifications. But this objective lens, so it's a rotating mechanism. We can rotate it and we can bring a different objective lens over there, right? And that objective lens can have different magnification. So when I want to go ahead and look at a image with 100 times magnification, I can simply use my objective lens to magnify it, right? Below the objective lens, you will have your stage. Stage is the area of the microscope on which you are going to be placing your glass slide. We have okay. It's the stage is the portion of the microscope on which you're going to be placing your glass slide. Okay. After placing your glass slide on the stage, you can move the stage up, down, left, right. So you can move it left, right, sideways to, of course, make sure that you can bring the image right under the lens. And secondly, you can then use a focus. So there are two focus in the microscope. Okay. We have a coarse focus and a fine focus. I'll show you Hold on, I'll show you an image. The course focus is going to allow you to move the stage up, down really quickly, right? So it's going to allow you to focus really quickly, and it's going to move the stage up and down, and it moves it fast. Now, one thing you guys have to be careful for, this is just a tip for your paper 3 exams, that whenever you are asked to focus on a slide, just be careful when you're doing course focus, because sometimes the stage moves so quickly that you don't realize it and you end up breaking the slide. So you have to make sure that you pay attention to how you focus it. Then you have a fine focus. Fine focus moves the stage in very small increments, right? So use the coarse focus to bring the image, make it a bit clearer. And when the image is somewhat clearer, then you use a fine focus to make it completely visual for you, completely visible for you. Okay. Below that, this is the below that we have this setup. You can't really see this. Below this, we have a light source that is going to be present at the bottom. We have a light source. And above the light source, we have a condenser. The condenser condenses the light, right? It focuses on all the light on our slide so we can visualize the image. This is what a light microscope, actually the different parts of the light microscope are. Now, when we talk about our light microscope, so there are some obvious, really clear advantages to light microscopy, right? I mean, first of all, this is relatively cheaper compared to other types of microscopes. Light microscope is cheaper. You can easily use it in school laboratories, in hospital laboratories, anywhere at all. It's easy, it's small, it's portable. You can simply carry it from one place to another. And one second. You can easily carry it from one place to another, and you can also visualize live cells, right? Live cells meaning, let's say I want to visualize bacteria. Using live microscopy, I can actually visualize a live bacteria. I will be able to see them moving, right? That's a significant advantage that light microscope gives you over other types of microscope. So a light microscope allows you to visualize live cells. It is relatively cheaper, it is portable, it is small, and it can easily be used in wide variety of settings. But what is the disadvantage of light microscope? One of the major disadvantages of light microscope is that it has low magnification and low resolution. What is the difference though? What is the difference between magnification and resolution? Magnification is how big a cell is looking or how big an image is looking compared to the original size. Whatever my original size is, compared to it, how big is my image? That is magnification. Resolution is how clearly you can differentiate between 2 points. So if I have two structures in my cell, how clearly can I differentiate between them? Any microscope that will have high resolution, so you will clearly be able to see that, oh, this is a distinct structure and this is a distinct structure. If you have a microscope that has low resolution, then you will not be able to distinguish between them. So resolution is the ability to distinguish between 2 points, between two objects. Magnification is how much time or how much bigger is the image compared to the actual object. And the formula for magnification is the same that you've done in IGCSE. That is not going to be different, which is magnification is equals to image size divided by actual size of the object. right? That is what magnification is. Okay. Now, when we go ahead and we talk about it, so how does um, how does the process actually work? How does a light microscope actually work? So a light microscope works like this. We have visible light that you and I can actually see. That visible light is going to that visible light is going to pass from the bottom, from our light source, to over here, that this is going to be our slide. On our slide, we have our tissue piece. Whatever we're trying to see the cheek cell from a cheek. So the uh, structures in the inside the cell will scatter the light, right? They will absorb the light, they will scatter the light, and that scattering of light will travel up to the eyepiece, and in the eyepiece I will be able to see what the structure actually is. So when we go ahead and we talk about it, right? So this is how it actually is going to be working. So those structures that are large enough to scatter the light waves, because those structures that are large enough to interfere with the light waves, those I will be able to see on the light microscope. Those objects that are too small, they're smaller than, for example, the smallest we have is 400 nanometers. So those structures of the cell that are smaller than 400 nanometers, they will not be visible to me. Why? Simply because the light is going to pass from between it, instead of scattering, instead of the structure interfering with the light will simply pass through and I will not be able to visualize it, right? So that is the problem with our, like so if I have a particle that is smaller than the wavelength, I will not be able to observe it. An object or a structure has to be large enough to interfere with the visible light. Then, yes, I will be able to see it using a light microscope. So, what do I do? How can I visualize those structures that are not visible with a light microscope? How do I visualize those structures that cannot be seen? Those structures that are too small. Those structures that I told you, we call them ultrastructures. To visualize them, we need an electron microscope. How does an electron microscope work? So an electron microscope works using electrons. You guys have studied in physics and in chemistry in IGCSE that electrons are particles. But quantum physics has taught us, and this is something you guys will study in your physics, right? When you guys do, if you guys do A-level physics, you'll study this. Electrons can also travel as a wave. Electrons can also travel as a wave, and the wavelength of electrons is very, 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 very small. So all those structures that cannot be visualized using a light microscope, we can see them using an electron microscope. Because the electrons are the wavelength of electrons is extremely, extremely small, right? So when we talk about electron microscope, how does it actually work? So you don't need to know this entire diagram. You don't need to know the working of it. What you need to know is this. First of all, when we talk about a light microscope, so in a light microscope, we have to use something known as dyes or stains, okay? In a light microscope, we use stains. What are stains? When you talk about your cell, so cell does not have any color on it whatsoever. If I take a simple cell and I put it in a microscope, you will not really be able to distinguish between the nucleus and the cell membrane and the cytoplasm and the mitochondria, simply because it's transparent. For me to be able to actually see something on the cell, I will have to stain it. I will have to add a dye to it. So we have variety of different stains that are available. that we can use in light microscopy to be able to visualize, to be able to actually see what the cell is, what cell is present in front of me. For that, I need to have states. So they're chemical compounds, they're dyes, they're colors that I can add to be able to see my cell. On the other hand, in electron microscope, we use heavy metal dyes. We use heavy metal dyes. Heavy metal dyes are used to stain the cells. So for light microscopy, it's generally chemical compounds. For electron microscopy, we have to stain the cells with heavy metals, right? And what happens that when the electrons, so we have an electron beam. We literally are shooting electrons on the cell. So, and what is on top of the cell? Heavy metal. I just covered the cell in metal. I dyed the cell with metal, like gold. So what's going to happen? That uh, um, um, when your metal is going to become hot, right? The metal of the cell is going to become hot. What's going to happen? The metal that is staining the cell is going to become hot. Some of the electrons are going to escape. They're going to become delocalized. They're going to escape. And as they're going to escape, they are going to turn into a wave. And we will be able to observe that wavelength. So different structures will be emitting electrons of different wavelength, and then we can use that to visualize our cell, to be able to see our cell. Now, when we talk about electron microscope, we have two types of electron microscope: a transmission electron microscope and a scanning electron microscope. A transition electron microscope allows you to see the inside of the cell, to see the cross section. We call it cross section. It allows us to see the inside cross section of the cell. If I want to see the inside of the cell, I will be using a transmission electron microscope, right? On the other hand, if I want to see the surface of the cell, I will be using a scanning electron microscope. This is a pollen grain. So you can see the surface of the pollen grain, right? So it gives you a three-dimensional image. Can you see it's 3D? It gives you a three-dimensional image, and it is allowing you to see the surface of the cell. Transmission electron microscope allows you to see the inner structure of the cell, right? The advantage of this scanning electron microscope. What is the advantage? The advantage of scanning electron microscope is, first, you are able to visualize the surface of the cell. Secondly, you are able to uh, you are able to see a three-dimensional appearance, right? But what is the disadvantage of scanning electron microscope? Low resolution compared to this one. Compared to transmission one, it has low resolution, right? But this was just about this scanning electron microscope. You don't need to go into details of any further detail than what I just told you. Now, let's do an overall advantage and disadvantage of overall electron microscopy, not specifically transmission and scanning electron. Overall, what is the advantage of electron microscope? One huge advantage of electron microscope: really high magnification and really high resolution. Extremely high magnification, extremely high resolution. Without a scaling electron microscope, a lot of what we know about the cell, we would not know. It is a huge advantage. It allows us to visualize really small structures inside the cell, because of the fact that it has a really high resolution and it has a really high magnification. Really good, amazing. But it has a lot of disadvantages. Of I bet if I asked you guys a few disadvantages, you guys could tell me straight away. You would say it's expensive. It's expensive. Electron microscope is very expensive. you need to keep it in a very special area. It's not portable. It's a huge machine. You need to keep it in a very special area. You need to hire special staff, specially trained staff to maintain it, and specially trained staff to use it as well. So your staff has to be highly trained. It is extremely expensive. You need to be able to have a really huge room available where you can keep the electron microscope, and you cannot use it to see live cell. You cannot use it to see living cells. Why? Because inside a electron microscope, it's a vacuum. There is no air. Inside, not outside, obviously. Inside the machine, there is no air. It's all vacuum. So of course, you cannot use it to visualize any living cells. That's why I told you for light microscope, we can use it to visualize live cells as well. That's an advantage, a big advantage. But electron microscope, we cannot visualize live cells. It's really huge, really costly. You need specially trained staff to be able to maintain it. But it gives you really amazing resolution and really good magnification. So that is the advantage of your electron microscope and your light microscope. Does anybody have any confusion so far? Anuj, Khadija, does anybody want to ask anything?
No, miss.
Okay. Okay, so there's, you guys, when we talk about our light microscopy, right? When we talk about how we're going to use a microscope to visualize a cell, so one new thing that you guys need to know and use for your calculations in your A levels is you guys need to know how to use a eyepiece graticule. I will tell you what that is. I will tell you what it is. One second, let me open up and let me tell you. So let's say hold on, I'm just opening it up. Okay. So let's say that I come up to you, right? And I say that you need to, I am putting a microscope in front of you. Okay. The microscope is open in front of you, and what you have to do is that you have to determine the actual size of the cell. Now, what are you going to do? You're going to say, Miss, how am I supposed to find out what the actual size of the cell is? I mean, I have the microscope in front of me. How am I supposed to visualize, or how am I supposed to tell what the actual size is? How do I measure? I mean, I can't use a ruler. I can't use a scale. It's a cell. How am I supposed to tell you what the actual size is? So for that, we use something known as an eyepiece graticule. What is the eyepiece graticule? A graticule is a scale. I'm showing you guys a picture. Wait 1 minute. A graticule is a scale that is found built into the eyepiece. The eyepiece actually has a scale inside of it, right? That scale is known as the eyepiece graticule. So you can use the eyepiece graticule to measure the actual diameter of the cell. What is the real diameter of the cell, right? You can go ahead and visualize that by using an eyepiece graticule. But there's a teeny tiny problem. The teeny tiny problem is that the eyepiece graticule cannot be 100% trusted. Why? Because the eyepiece graticule changes with magnification. So what you and I need to do is that before we can use the eyepiece graticule for our calculations, we need to calibrate it, because this is what it looks like. Okay. This is the eyepiece graticule. This, you see, it's from 0 to 100. Okay. It is from 0 to 100. Now, what's going to happen that this eyepiece graticule, so it's a built-in scale inside my eyepiece. When you will look in the microscope, you'll be able to see the built-in scale. Now, there's one teeny tiny problem. Like I said, the eyepiece graticule is not reliable. The measurement is going to change if you change the magnification. So what can you and I do to make it so that we can get accurate measurements of our cells? We need to calibrate our eyepiece graticule. How can we calibrate it? The way we calibrate it is like this. We have another scale, and this scale is known as stage micrometer. How am I going to do my calibrations? Look, let's read the question. You are going to get these questions in paper one. Paper one has these type of questions inside of it. A student used a stage micrometer scale to calibrate an eyepiece graticule. Okay. The diagram shows the view of both the stage micrometer scale and the eyepiece graticule seen by the student. The divisions on the stage micrometer scale are 0.1 millimeter apart, meaning that if I take a look at this, the stage micrometer, so the divisions are 0.1 millimeter apart, meaning that from here to here, it's 0.1 millimeter. Now, so the student removed the stage micrometer cell and viewed a slide with blood cells on it. The same lens is per use, so the magnification remains unchanged. That's very good with me. The student measured the diameter of one of the white blood cells on the slide using eyepiece graticule and recorded that it was 8 eyepiece units. What is the correct diameter of this white blood cells in micrometers? What are we going to do? First thing is first, we need to know what is the value of one eyepiece graticule, right? One simple division, one division of my eyepiece graticule. What is the value? How many millimeter is it? I first need to know that. Before I can do anything else, I need to know one division, one small unit, one small line on my eyepiece graticule. What's the value? To do that, let's calibrate our eyepiece graticule. So you can see if I match the lines, look, this is the start of my one division or one unit on the stage micrometer. So let's map. So one division or one unit on my stage micrometer is equal to 40 eyepiece units, right? One division on my stage micrometer is equal to 50 minus 10 is 40. 40 of the eyepiece graticule, right? Now, what we are going to go ahead and do is we're going to do a simple division. One of the stage micrometer is equal to 40 of the eyepiece graticule. So one of the stage micrometer is 0.1 millimeter. So 0.1 millimeter is equals to is equal to x, right? And you can go ahead and you can do your calculations. Hold on, let me just pull out the calculator. Give me 1 minute. So, okay. So 1 micrometer division is equal to 40 of the eyepiece graticule, right? So that means 40 eyepiece graticule is equals to 0.1 millimeter. So if 40 eyepiece graticule is equals to 0.1 millimeter, so one eyepiece graticule is equal to this. And then we can solve and we can find out what is the value. How many millimeter is one single unit or one single division of the eyepiece graticule. So what have we done so far? One division of the stage micrometer is equal to 40 divisions on the eyepiece graticule. So that means 40 divisions is equals to 0.1 millimeter. So if 40 division is equals to 0.1 millimeter, one division of the eyepiece graticule is equals to x. And then we are going to go ahead and solve it for x. So what is going to be our answer? So let's go ahead and do our calculations. We are going to go ahead and do 0.1 divided by. So we get 0.0025, meaning that one single division, one single division on the eyepiece graticule is equals to 0.0025. Making sense so far? Khadija, Anuj, is this making sense so far, what we're doing so far?
Yes, miss.
Are you guys
Yes, miss.
completely sure? Completely clear? Okay. So simple, one small division on the eyepiece graticule is equals to 0.0025. Now, they said the red blood cell was 8 eyepiece units. So let's multiply by 8. What do we get? If we multiply by 8, multiply by 8, we get 0.02, right? So we are getting our answer as 0.02. Now, the answer you and I have just gotten is in millimeters. They're saying, what is the answer in micrometers? Now, for us to go ahead and do that, we first need to know what the conversions are. So remember it like this. This is how I want you guys to remember it for all your sciences subject, right? We have got meters. If I want to turn meters into millimeters, I will divide it by 1,000. If you want to write it down, you write it down as 10 raised to power negative 3. If I want to turn my meters into micrometers, I will do into 10 raised to power negative 6. Then after micrometers, we have nanometers. Nanometers in 10 raised to power negative 9. After nanometers, we have picometers. Picometers is 10 raised to power negative 12. Okay. And what is the difference between all of them? 1,000. If I divide millimeter by 1,000, I get micrometers. If I divide micrometers by 1,000, I get nanometers. If I divide nanometers by 1,000, I get picometers. So to divide millimeter into micrometer, I will divide by 1,000. Micrometer into millimeters divided by another 1,000. Another 1,000, right? So that's basically what I'm going to be doing. You're simply going to go ahead and keep on multiplying it or dividing it by 1,000, depending upon what you're doing. Are you going small to big or big to small? You will multiply or divide it by 1,000. Right? So in this case, we are trying to convert millimeters into micrometers. So we are going from big to small. Whenever we go from big to small, we multiply. I'll multiply my answer by 1,000. So I will get it as 20 micrometers. And our answer will be option D. Right? I'm going to show you guys a few more questions on how we can solve this. Okay, by the way, just for information, above meter, we have kilometer, which is 10 raised to power 3. Then we have mega, which is 10 raised to power 6. Then we have giga, which is 10 raised to power 9. And then we have tera, which is 10 raised to power 12. So you guys need to remember these suffixes. We call them suffixes, okay? So milli means 10 raised to power minus 3, anything. Millimeters, milligrams, any unit at all, milliseconds, take any unit that you want. But if you put a milli to it, 10 raised to power minus 3, micro is 10 raised to power minus 6. So these are the conversions that you guys need to know. Okay, so that is how we're going to solve this question. Let me open up another example question. This is making sense, right? No confusion in this, guys, how we did this question.
No, miss.
Okay, hold on, let me just open up another example for you guys. Okay, take a look at this. The diagram shows a stage micrometer with division 0.1 millimeter apart. So what is the value of one division? 0.1 millimeter. The area of the field of microscope can be calculated using this formula. So formula is given to us. A student calculated the area of the field of view of microscope using the information provided. Which answer has been rounded correctly to an appropriate number of significant figures? Before we can give that answer, we first need to calculate our value, right? Now, how are we going to calculate the value? Before we can do anything, we need to calibrate our eyepiece reticule. How are we going to calibrate our eyepiece reticule? We are going to match one division of the stage micrometer with our eyepiece reticule. So one division of the stage micrometer is equals to 40 of the eyepiece reticule. Fine. Now, so that means 40 of the eyepiece reticule is equals to 0.1 millimeter. Again, we will solve it using cross multiplication. 40 eyepiece unit is equal to 0.1 millimeter. So 1 eyepiece unit is equals to x. So again, we are going to go ahead and do our calculations. 0.1 divided by 40. What do we get? 0.0025. So 1 eyepiece unit is equals to 0.0025. Now, the formula is pi r square, where I is what? R is what? R is radius. We know that. So radius is going to be from 0 to 50, right? To the center of the circle. So 0 to 50. So multiply this by 50 to calculate our radius. So I'm going to multiply this by 50, multiply by 50, and that gives us 0.125 millimeters. Remember, units are really important. 0.125 millimeter. Then we are going to use our formula pi r square. Pi is 3.142. So let's do the calculations. Answer squared and then multiplied by 3.142. So we get 0.049 millimeter square. Right? Now, again, let's continue with our calculation. So now we need to give the answer. They're saying which one of this is rounded correctly to appropriate numbers of significant figures. So we have to tell them which one is rounded off correctly. So, okay, this option A matches our answer. We are getting the same answer, right? But this is not rounded off appropriately. We generally do three significant figures. Okay, so 1, 2, 3. This should have been 0.05. This is not the correct answer. When we are going to go ahead and talk about it, okay, let's convert this, or let's change this to a micrometer. If I change this to micrometer, I'll multiply by 1,000. And if I change this by multiply this by 1,000, I'll get 4.909 micrometer. And if I round it off correctly, it will become 4.91 micrometer, right? So answer is option D. Making sense? Is this all clear? Is this making sense, guys?
Yes.
Absolutely sure. No confusion regarding how to solve the IP square reticule, Aruj? Aruj, all clear? I hope it's clear how we can solve the IP square reticule questions.
Yes, miss.
Okay. So that is how we calibrate the eyepiece reticule, and that is how we go ahead and do our calculations related to our object. Now, when we go ahead, so that was what you had to know about. That is what you know about our microscopy portion of the topic. Now that we have covered the microscopy of portion of the topic, let's talk about the actual thing that we have to study, and that's the structure of the cell. Now, let's start from the center of the cell and slowly go outwards. So at the very center of the cell, we have the nucleus. Let's study about the nucleus. Okay. So when we talk about the nucleus, what points are you guys expected to know regarding our nucleus? Now, point number 1. Let's first talk about their physical characteristics. Okay. Nucleus is surrounded by a double membrane that is known as nuclear envelope. Right? So it is surrounded by a double membrane that we call as nuclear envelope or nuclear membrane. But nuclear envelope is the common term that we use. So it is surrounded by a double membrane. And when we talk about it, the double membrane means that it has got two parts. We have an inner membrane and an outer membrane. It looks like this. Right? We have an outer nuclear membrane and we have a inner nuclear membrane. Between these membrane, we have small openings. These openings are known as nuclear pits. What is the function of nuclear pits? It allows exchange of proteins. It allows exchange of substances from the nucleus to the cytoplasm. So for example, if you guys remember your IGCSE biology, messenger RNA, right? Messenger RNA, and if you guys don't remember, it's fine. We have the whole chapter on it coming up later on, okay, this year. So messenger RNA, for example, is really important for making proteins. So it is able to leave the nucleus and go into the cytoplasm using your nuclear pits, right? Then along with that, what else do we, nuclear pits actually not exactly pits, we call them nuclear pores. That is a better term. We have nuclear pores. Use the term nuclear pores. That's a better term. So we have nuclear envelope in which we have an inner membrane and outer membrane. And we have spaces or openings between the between the envelope, which we call nuclear pores. And what is the function of nuclear pores? To allow exchange of material like messenger RNA to travel between the nucleus and the cytoplasm. Now, along with this, what else is present inside our cell? Of course, we have DNA. But DNA is found in the form of a coiled thread that we call chromatin. Okay, so DNA is not found as DNA. It is found as a thread-like structure called the chromatin. And what is another structure that is found within the nucleus? We have a really darkly staining area, an area that is really, really darkly staining. And that darkly staining area is known as nucleolus. The function of the nucleolus is to make ribosomes. Ribosomes are what? They are small proteins that we need for making proteins, right? We need them for making proteins. We need them for making ribosomes, and to be very specific, they make ribosomal RNA. Ribosomes are made up of RNA. We will study that when we get to the topic. So we specifically need the nucleolus to make ribosomes. Very specifically speaking, we needed to make ribosomal RNA. So we have our nucleolus. And of course, we have our DNA in the form of chromatin, right? That is what the structure of nucleus is. Now, when we talk about, let's move a little bit out of the nucleus. Let's go a little bit out of the nucleus. When we go a little bit out of the nucleus, we reach another structure of the cell that is known as endoplasmic reticulum. What is endoplasmic reticulum? Endoplasmic reticulum is a membrane. It's like a network. It's like a system, a network of membranes, right? Flattened membranes, flattened compartments. It's like a network of flattened compartments, and we call those compartments sacs. Okay. So they are these network of flattened compartments that we call them sacs. So we have got network of flattened sacs that are just spreading throughout the cell. Now, one thing, this endoplasmic reticulum is continuous with the outer membrane of the nucleus. What does it look like? So imagine this is my nucleus. This is the outer membrane of the nucleus, right? So continuous with the outer membrane of the nucleus, I will have this extensive, huge network of these sacs just connected like this. You know, it's going to be like a huge network of these flattened out discs, flattened out compartments. And this is basically known as your endoplasmic reticulum, right? This is what is known as endoplasmic reticulum. So, and it's continuous with the outer surface of the, it's continuous with the outer membrane of the nucleus, right? So when we talk about endoplasmic reticulum, fine, I've told you what endoplasmic reticulum is, but what does it do? Why do we have it? What's the function of it? What's its relationship? What is it there for? So to understand this, we need to know that we have got two types of endoplasmic reticulum. We have rough endoplasmic reticulum rough endoplasmic reticulum has small dots all over it. And I'll show you a real picture of it as well. It's got these small dots all over it. What are these small dots? These small dots all over it are ribosomes. And that should tell you what is the function of the rough ER. The rough ER or the rough endoplasmic reticulum is covered in ribosomes and it has the function of making protein. So your proteins are going to be made in the ribosomes because it is attached to the ribosome. This is where the proteins are going to be made, right? Along with that, we have another type of endoplasmic reticulum, smooth endoplasmic reticulum. Smooth endoplasmic reticulum does not have any ribosomes. It's not attached to any ribosomes. It has a very smooth appearance. Similar flattened disc, flattened compartment, similar in that manner, but this does not have any ribosomes. What is the function of the smooth endoplasmic reticulum? To make lipid. So yours, for example, any lipid-based product, lipid-based hormone like steroids, where will they be made? They will be made in your endoplasmic reticulum. When you talk about cholesterol, endoplasmic reticulum. When you talk about hormones like your testosterone, estrogen, progesterone, these are all made in the smooth endoplasmic reticulum. So we have, and this is what it looks like. Let me show you guys 1 second. So this is the nucleus on the inside, right? And see how it is continuous. Can you see how it is continuous with the outer membrane of the nucleus? This is endoplasmic reticulum. This is how it looks like in real. This over here, this structure that you see, this is your nucleus over here. This is the outer nuclear membrane. And these discs that you see, this line, this line, this line, this line, this line. Can you see these small black dots? One dot over here, a dot over here, a dot over here. Can you see the small black dots? They're all ribosomes. So this is the endoplasmic reticulum. Because it has ribosomes on it, this is specifically the rough endoplasmic reticulum. Any confusion? Anybody wants to ask anything so far?
No.
No.
After the rough endoplasmic reticulum, we have another structure that we call Golgi body or Golgi apparatus. Golgi apparatus or Golgi body looks like flattened stack of dishes. Like I've taken a stack of dishes and I have placed them one on top of the other. It looks like a stack of dishes, right? So your Golgi body, we can call it Golgi apparatus, Golgi body, Golgi complex also. You'll see this. So it is a stack that endoplasmic reticulum was a network. This is not a network. This is flattened stack that have been stacked. Right? You are going to see a lot of vesicles associated with it. It'll seem like, oh, there are vesicles breaking off from, different portions of the Golgi body. There are vesicles coming off from different portion of the Golgi body. They're joining the Golgi body or they're breaking and flowing away. Both things you will see. These vesicles are known as Golgi vesicles. What are they? What are these vesicles? So remember how I told you that the endoplasmic reticulum makes proteins and lipids, right? Now, those proteins and lipids have to undergo further modification before they are ready to do their job. Okay? That further modification is going to occur in where? It's going to occur here, inside the Golgi apparatus. Golgi apparatus or Golgi body will further modify the proteins or lipids to make them into functional structures. So endoplasmic reticulum is making the base, doing the basic portion of it. But before it can actually give you a functional hormone, for example, it has to go to Golgi vesicles. So what's going to happen that a vesicle containing protein will break off from the endoplasmic reticulum and then fuse with the Golgi body. There, it will undergo further modification, and once prepared, it will bud off or it will break away from the Golgi body as a vesicle containing the finished product. So first, what's going to happen? I have my endoplasmic reticulum over here. So a vesicle containing that protein will come from the endoplasmic reticulum and it will go to the Golgi body and it will fuse with the Golgi body. Over there, the protein is going to undergo modifications until it turns into the complete protein structure that you and I are looking for. Once that modification is done, once we have the complete structure, the vesicle will again break away from the Golgi body. It will move away from the Golgi body. And now this vesicle has the finished product. It has the finished hormone. So now it can go to the cell membrane and it can be released into the bloodstream or wherever it has to be released. So that is what the Golgi vesicle does. So it collects, processes, and sorts molecules, right? It gets them ready for transport. And then those vesicles, they can either stay within the cell and do their function inside the cell if that's their job, or they can go out of the cell, whatever needs to be done, whatever the function of that protein is, right? So endoplasmic reticulum, the vesicle will move from endoplasmic reticulum to Golgi body. Golgi body will make additions, will make modifications, get it ready to its final structure. And once it's ready to final structure, it will be again released as a vesicle, and then it can stay within the cell or it can be secreted out of the cell, removed from the cell, depending upon whatever the structure is. What kind of modifications it does? So it can add, it can add stuff to the protein, like it can add sugars, it can add sugar, it can add carbohydrates to the to the to the protein, right? We call it glycosidation. We call it glycosidation. We're taking a protein and we're adding sugar to it. We're adding glucose component to it. So glycosidation, right? We can add sugar to it, right? And we can add sugar to proteins to give us a structure that we call glycoproteins. We will study about glycoproteins, by the way, later on. So we can combine to give us glycoproteins. That is one thing that we can do, right? Now, along with that, what else can we go ahead and do? We can, another thing that it does is, it will remove the first amino acid. So there is the first amino acid in every single protein. The first amino acid is always methionine, always, always, always. And I'll tell you guys why when we do the topic of this protein thing, right? So first amino acid is always methionine. So it will remove that first amino acid. It will remove methionine so that we will have a functioning protein, right? Then along with that, it is going to, so this is an example of some of the structure it's going to do. It's going to add components to the proteins like sugars. It will remove the first amino acid to make it into a functioning protein. So all of these modifications are going to be done by what? It's going to be done done by the Golgi apparatus, the Golgi body. And if I show you what it looks like in real, so this is what it looks like in real. This structure. So this, see how it's like, it's not connected to each other. It's not a network. It's flattened disk that is stacked on top of each other. And you have, see these are all vesicles that are either fusing with the Golgi body or breaking away from the Golgi body after the proteins have been modified. Making sense? Does anybody want to ask any questions so far?
No.
Aruj, Khadija, all clear?
Yeah.
Are you sure
Yes,
nobody has
miss.
any question? Clear? Okay. All right, guys. So we will go ahead and stop it over here for today. And then we will continue studying on the cell in the next class. All right.